So our next presenter is Linda Beckett, and Linda is from Purdue University, and her presentation is entitled, "What Goes Around Comes Around, Carbon Cycling in the Liver Supports Milk Production."
Thank you for that introduction, Dr. Borman. Good afternoon, everyone. Thanks for virtually turning in today. So when you think about what goes around comes around, you might literally think about a Ferris wheel or a merry-go-round, or you might figuratively think about the what goes around comes around phrase where regarding karma of what goes around might come around and bite you. But I think about carbon cycling in the liver of dairy cows and how that can support milk production. And so when we think about carbon cycling and how it can aid milk production, I want you to focus in on the idea of nutrient interaction. So how, for example, short-chain fatty acids can interact with glucose. And a good location for those interactions is the TCA cycle and the connection to gluconeogenesis in the liver. And so we know that the TCA cycle and gluconeogenesis produces these energetic currencies such as ATP, NADH, FADH2, and glucose. And these different energetic currencies can go to different tissues and organs in the cow, but specifically are going to head towards the mammary gland for milk production and milk component synthesis. And so my simplistic diagram is representing that. And the next slide we'll get into more of the biochemistry. But one of our main research problems or research focuses in my lab at Purdue is how do these nutrients interact at the TCA cycle and gluconeogenic side of how they interact at the TCA cycle and gluconeogenic side, and how that can support milk production and milk component synthesis. And so when we think about the TCA cycle, and I know this is a very complex diagram, but I want you to focus in on the general idea that we have these short-chain fatty acids, such as propionate and lactate, that we know are produced from dietary glucose fermentation in the rumen. And these short-chain fatty acids directly intervene into this TCA cycle at succinyl-CoA and pyruvate. And the black dots beside each intermediate denote the carbons in that particular metabolite. And the red carbon here is how we are able to track the movement of these carbons. And so like the previous presentation noted, the use of carbon-13, that's exactly what we use as well. And we're able to make these carbons heavier and track their molecular weight through the latter half of the TCA cycle and up towards gluconeogenesis. And I know this is an abbreviated version of gluconeogenesis, but this is one potential way that this carbon could move. Another potential way is this carbon could just go through the TCA cycle rounds and continue in the cycle for a numerous amount of rounds. That's still yet to be determined on our front. Also, this carbon label could be exported as CO2, or could even label the ATP that is our energetic currency. And so when we're tracking these carbons, we want to figure out essentially where they go, like I mentioned before, but also we want to know where do they come from. So another just more layman's terms research question is, where does the carbon come from and where does it go? And so there's a number of ways we can do this. And if you've ever worked with stable isotopes, you probably understand the expense of the stable isotope and also the labor of having to infuse it into an animal. And so we typically steer clear of whole animal experiments due to the labor and expense. But one way we're able to essentially still get the same amount and tremendous amount of data is by taking liver biopsies from our cows. And this is actually an example of a very large liver biopsy that we get. We normally get about half the size of that, but we're able to divide this liver biopsy into 20 to 30 milligram pieces and incubate those pieces in carbon-13 labeled media. So we would typically take these biopsies and incubate them in propionate and lactate labeled media. And we incubate them in the media for 2 hours. And then after some further processing of the samples, we're able to analyze them on a liquid chromatography tandem mass spec. And this allows us to identify all the different, mass isotopomers that arise during our carbon-13 labeling. And then we're able to essentially put our data into our modeling software so that we're able to quantify the carbon flux and carbon movement through the TCA cycle and up towards gluconeogenesis. And so when we're bringing this back to how does carbon cycling aid milk production, because I know I got into a lot of the biochemistry there. If we're able to understand, like I said before, where does the carbon stem from and where does it go, we're able to maximize our use of that carbon. So we're able to better feed cows in that regard. And additionally, we're able to increase the production efficiency of our cows. And this picture, I think, is following suit with my theme of what goes around comes around, if you get my pun there. And then lastly, we're able to maximize our use of this carbon for the transition cows. We know that during the transition period that the cows it's a very metabolically chaotic period for cows. And if we can ultimately maximize the use of those carbons and maximize the use of those substrates, then we're able to then these cows are able to be more healthy and enter into lactation, enter into lactation stronger. And so that's what we want to do with carbon cycling, is we're using the biochemistry to aid to increase efficiency of production and maximize animal health and well-being. And so with that being said, I'll take any questions, and I'd like to thank you for virtually tuning in. And my email is up here as well if you'd like to send any questions that way.